Part of the fun of this topic is that it brings together people from all walks of life. And you never really know who might have synesthesia. You might not even know you have synesthesia. Maybe I should ask you, how many of you are non-scientists or not really neuroscience? Yeah, great. Serving a good distribution. In fact, one of the people who I was surprised to learn who has synesthesia is the former dean of Harvard Medical School, Joe Martin. And Joe is an old friend, actually, from UCSF days. And he was very sad that he couldn't be here, because it's something that he's been quite passionate about understanding and loves to tell his story. So he volunteered to tape his story for us, and I thought it was a nice way to launch the afternoon session. So hopefully this will work, and we'll hear from Joe Martin, the former dean of Harvard Medical School.
Good morning, ladies and gentlemen. I want to welcome you to this conference, which I wish I could attend with you. However, another commitment took me out of the country, and I look forward on my return to hearing more about some of the ideas that were presented at this conference. My story is a personal one, and I've entitled it The Imaginary World of Synesthesia. And I'd like to start by just reflecting on what some have said are important introspective issues as we try to understand how the brain works. It's a picture of me here with C. Miller Fisher, who some of you may recognize by name or by face as probably the most important contributor to the world of stroke pathology and clinical medicine over many, many years. This is a photo taken with him at age 96, when he was still active writing and talking about his experiences. He made the point often to us as residents, as junior faculty, that it was important as you learned about patients in neurology, which is my field, to try and understand exactly what they were saying by internalizing some of it into your own experience. And I always took this as a gift of knowledge that I think has helped all of us who are clinicians to try and interpret in a better way what our patients are really trying to say. So I'm going to follow his example and do a bit of reflection. Before that, let me just show a slide that illustrates another hero of mine, David Hubel, who also I found in life often reflected on his own personal subjective experiences while he was carrying on daily activities. And this is a slide from Nature in 1997. He, of course, was a Nobel laureate. When I get up at night, dark adapted over many hours, there is just enough light for me to make my way around, but too little to allow me to find small objects. By cupping a hand lightly over one eye before turning on the lights, this nuisance can be avoided, because when the lights are turned off again, dark adaptation is fully preserved in the covered eye, although the other eye is temporarily completely blind. A good way to find your way to the bathroom at night. My story begins back some years in pre-med, when a professor of psychology addressing a class of 200, 250 people said, "How many of you imagine numbers in lines in space?" And I vividly recall perhaps 5 or 6 students in the class putting their hands up, of which, of course, I was one. So I knew exactly what he was referring to, and he went on to say, "The rest of you won't have a clue what I'm talking about." Well, the first description of number lines came from Sir Francis Galton in 1880. And I learned of this paper from Dr. Ramachandran, who many of you know well and from his writings, when I was visiting him in San Diego one time. Told him about my own propensity toward this, and he referred me to Sir Francis Galton's paper in Nature. As you can see here, I have lately been occupied in eliciting the degree and manner in which different persons possess the power of seeing images in their mind's eye. The various ways in which numerals are visualized is one that is curious and complete in itself. Not only visualize their numerals, but also invariably assign to each of them a definite place in their mental field of view. And this was, in fact, from that paper, showing 3 subjects in which the number lines are illustrated totally random in terms of their differences one from the other. And so it was that I could draw a graph of my own, and I will show that in a moment. The question of where such a capacity might be localized is reported in a case published in 1950, where there was a disturbance of this capacity in the case of brain injury. And they point out again, as Galton had, that among persons who visualize numbers clearly, there are many who notice that the image of the same figure invariably makes its appearance in the mental field in the same direction and at the same distance. And so the notion springs forward that there is a tendency to see the same visualization from time to time over months, years, decades, and so is the case with my own, which I'll show in a moment. What they go on to report is that in this particular patient, there was a right parietal lobe infarct as a result of the injury, and that this patient had lost the spatial synesthesia experience. So here's my number line, starting with 1, progressing, as you can see, to 12, where it dips sharply to the right, and then a kind of tangled web going down to 100, over to 500, 1,000, and then forming almost concentric squares going on up to 1,000,000, as I've shown here. The number line, as far as I can recall, has been exactly the same from the first time I was aware of it. And of course I was puzzled at why everyone didn't in fact see numbers in this fashion. I want you to look particularly at this little block I've outlined in red, which shows the numbers going up from 6 to 12 and then over to 20, because that will recur in the next set of photos or of diagrams that I show to you. Not only do numbers appear in space in terms of the numerical sequence, but I've always also put numbers to dates. And you can see in this particular illustration again, going from 1800 up to 2010 and beyond, that within my view of items historically, within my imagination about when historic events occurred, they invariably appear on this number line, making it really quite easy to recall spots along the line and to remember historical events, which I will illustrate in the next slide, where you can see that if I attach identities to this line, whenever I think of the War of 1812, it appears exactly on that point in the line. The Civil War, World War I, interestingly, is a very flat line. And again, I think you'll begin to see an objectivity of the way the line goes that I'll illustrate momentarily. World War II, moved to Boston. That seemed to be a big event. Then a decline, and then a decade in San Francisco, and then back again from 2000 to 2012, the same as it was from 1900 to 1912. And again, you can see this kind of repetition of a formed sequence of numbers that I experience. The number lines are not the only way in which the visualization identity occurs. This is an example of the calendar, which I always think of in a circle, as shown here. The days of the week are linear. And if I move on to some other examples, you will see how remarkably variable these various components of synesthesia is. Just a further moment about the spatial organization. I think that I have, in terms of right parietal function, a quite good sense of maps, of directions in a three-dimensional space that we move in, drive in, and a sense of where things are, and a recollection and memory of those maps, if you like, that gives me a kind of GPS sense that I use all the time. A few years ago, I bumped into this gentleman, George, age 79, and we got to talking about these things, and he relayed to me this remarkable example of his own use of letters and of months of the years, and in this case showing colors that attended to them or with them. I have no color perception at all. Mine's simply black and white. But you can see if you go through the letters of the alphabet that each one of those letters has a distinct color. And you can see that if he does the months of the year, each of those months also has a distinct color. Now, the remarkable thing about this was that he told me that his daughter similarly has this kind of sense. And here's an example in her, from her path with respect to months in space and colors in space, a very different pattern from her father, but equally vivid. And when asked to do numbers from 1 to 10, she showed again a separation of each individual digit with a number with a color attached to it, and the letters of the alphabet similarly. This is important because it's well known that synesthesia does have a familial or likely genetic basis in some families, and a particularly important paper to this issue was published in 2009 from Oxford in England, in which genetic studies were done across 43 multiplex families with a small number of microsatellite markers, and they suggested linkage to 4 chromosomes, although the work clearly isn't sufficiently detailed to give any common sense of where a genetic locus might reside. Well, in conclusion, this topic of your day is really quite remarkable in terms of those in the literature who have referred to it. Oliver Sacks, who sadly passed away just a few weeks ago, said, "For a synesthete, there is simply an instant conjoining of sensation. This may involve any of the senses. For example, one person may perceive individual letters or days of the week as having their own particular colors. Another may feel that every color has its own peculiar smell, or every musical interval its own taste." And I particularly like the quote from Kandinsky: "The violins, the deep tones of the basses, and especially the wind instruments, at that time embodied for me all the power of that pre-nocturnal hour. I saw all of my colors in my mind. They stood before my eyes. Wild, almost crazy lines were sketched in front of me." So I think your Congress today, which has an extraordinary group of speakers, I've read many of their papers and works, and I am deeply regretful that I'm not able to join in with you today. But I think a window into brain development is illustrated here with the kinds of topics that I'm sure you'll be discussing, and that hopefully at the end of the day you will have some new sense about. What types of synesthesia are there? How do we know for sure that this is authentic and not just somebody's wild imaginations? When does it start? What are the possible mechanisms? Genetics, pruning, cross-modal inhibition, and what does it signify in respect to some of the clinical syndromes or creative genius that we acknowledge in some? So I wish you the very best today, and I look forward to hearing about the conference. Thank you.
Okay. Well, I want to thank Dean Martin for his personal story. It's always very interesting to hear. Having received my degree from his own hands when he was the dean at UC San Francisco, I was a little disappointed about his number line, depressing in his San Francisco years. But I do thank him for putting a shout out to our next speaker, in fact, about the genetics and potential genetic basis of synesthesia. Simon Baron-Cohen is a professor of developmental psychopathology at the University of Cambridge and a fellow at Trinity College. And he's very well known for his work on autism. I think he directs the Center for Autism Research there. He's a co-editor of Molecular Autism, and has published well over 400 papers. So I'm sure you've come across his work in one form or another. And we are really happy that you could make the leap over the pond to join us today. Thank you for coming.
First of all, I want to thank Taco and Daphne for inviting me to this meeting. It's a special meeting for me at lots of levels because, as you'll hear, I work in the field of autism research, but way back when I was a graduate student, I also worked in the field of synesthesia and kind of left it behind. Although over the years, people have said, let's just do one more study in synesthesia, but it was a very minor research interest of mine. And for a couple of decades, I really did think that although I had two fields of research, autism, which is a kind of major interest, and synesthesia, the kind of minor one, I thought they were completely disconnected. You know, sometimes you hope that two areas will connect. I think today we're going to hear quite a lot about possible connections between these two fields. So for me, it's one of those awakening moments. And I'm going to be talking also about prenatal steroid hormones, such as testosterone, and whether they might be relevant to both conditions. So starting off with acknowledgements, you can see a lot of names on this slide, but people who've worked with me on synesthesia going way back to the 1980s and on autism more recently. I'll just give you my definition of synesthesia, which is all about a perception in one modality being triggered by one in another, a sensation in another. And I think this is, it's argued, because in recent years the definition of synesthesia has become very stretched. Some would say diluted, so that sometimes you get reports of synesthesia that might be within one modality and may even be outside of the traditional sensory areas, like including personality, as was mentioned earlier. But here's my definition. But most importantly, I think what I've been interested in is so-called developmental synesthesia. That's to say, the kind of synesthesia that Joseph in the last video presentation was describing. He's had it all his life, as many of the previous speakers today have described from cases that it's automatic. People just assume it's just always how they've perceived the world, and that they assume everybody else perceives the world in the same way. So critically, it's not secondary to taking some kind of hallucinogenic drug or having some kind of brain damage or other mind-altering substance. So way back in 1987, I published this paper, and it was really an attempt to see whether synesthesia was real and could be subject to scientific inquiry. As Joseph said before, how do we know that people aren't just making it up? They may come up with all kinds of poetic accounts of their experience, and that was the case from probably the early 1900s way up till the 1950s and 60s, that we really just had a lot of anecdotal reports of people who had synesthesia with no way of validating that what they were saying had any reality. So what we published in 1987 was the so-called test of genuineness, and actually just reduces to a test of consistency. So we studied one individual who claimed to have synesthesia, and we gave her, as you can see, over 103 words. She had colors triggered by words. So we gave her a list of words, asked her to report the colors that each word triggered, and then without warning her, we retested her, and she was 100% consistent 8 months later. So we took consistency as evidence that this was real, in the sense that memory would be an unlikely explanation for such good consistency. We also had a control adult who, in this case, we asked her to make color associations to words, and we warned her that we were going to be retesting her, and despite the effort and the warning, she was only 17% consistent 2 weeks later. So consistency became a kind of definition of, if you like, diagnosis of synesthesia. We then kind of improved the test of genuineness by using these colored paint charts that you can buy in any hardware store, by asking people to pinpoint which color is triggered by, for example, a word or a letter, and then, of course, retesting them without warning so that we could get much more precision. But essentially, this has become the method, you'll hear from David Eagleman later, of identifying that this is a real phenomenon. Some people didn't believe that synesthesia was real until you could see some evidence in the brain. So we conducted the first imaging study back in 1995 of synesthesia using PET. So we blindfolded 6 people with coloured hearing, and they were played through headphones. They were played pure tones and words, and we saw activation in visual cortex even though they had no visual input. And so that gave us some confidence that synesthesia was real. You could see something in the brain. Of course, it doesn't really prove anything because you could get this kind of result just from pure imagination. We then used fMRI when that came along in 2002, finding activation in V4, when again people were blindfolded listening to words, synesthetes, but not in controls. So some evidence at the neurophysiological level of perhaps unusual connectivity in the brain, and this is a theme that I think has been running through the day. Other people have used DTI in synesthesia. This is one such example where you look at white matter and finding increased connectivity in various white matter tracts in synesthetes compared to controls. So again, some evidence of over-connectivity in the synesthetic brain. Joseph, in his video address, talked about the genetics of synesthesia, and we certainly saw evidence of synesthesia running in families, and you can see that this particular study in 2005 suggested that about 1/3 of synesthetes have at least one other family member who also has synesthesia. Well, that doesn't prove genetics because families share environments as well as genes. We conducted the first linkage molecular genetic study of synesthesia, which Joseph mentioned was conducted in Oxford. In fact, it was a collaboration between Oxford and Cambridge. I do have to say that. And all it was a linkage peak on chromosome 2, so it didn't pick out genes particularly that were associated with synesthesia, but it was certainly giving more confirmation that synesthesia was real at the molecular level. So the skeptics, who had never been silent all the way through, raising the question about does synesthesia exist, is it actually real? Some of them begun to be persuaded by a genetic association. So the advantage of coming late in the day is that I can comment on other people's comments during the conference. And so I've been amending my talk as other people have said interesting things. And this slide is really for Daphne, because just before lunch, I think you mentioned about perfect pitch being more common in synesthesia. Okay, but either way, what you can see here is a genetic study that I was a collaborator on, but it was reported by Peter Gregerson in New York, finding a genetic association with synesthesia and absolute pitch or perfect pitch. So that's kind of interesting. And we're going to kind of come back to this set of connections between perfect pitch, synesthesia, and autism, because you can see here that there's also been a study showing that people with perfect pitch have more autistic traits than musicians without perfect pitch. So an interesting set of connections between these three apparently separate diagnoses. So let me switch now to say a little bit about autism. Here's a kind of classic image of a child with autism playing alone, so that whole diagnostic feature of social difficulties, communication difficulties, but a love of patterns, as you can see. And traditionally, the diagnosis didn't really mention anything to do with sensory issues. So up until DSM-4, if you'd asked people, how do you diagnose autism, they would have said, well, it's right at the intersect between social communication difficulties and on the right, and repetitive behavior or narrow interests on the left, that if you have the co-occurrence of those, that adds up to autism if these features are interfering with your life. But DSM-5 changed all that. It's published in 2013 and now includes sensory issues as part of the diagnosis of autism, which suddenly means autism is getting a little bit closer to synesthesia. So we wondered whether synesthesia might be more common in people with autism, and published a study in 2013 to investigate this, because we reasoned that synesthesia is about 4% of the population, autism is about 1% of the population. If they were truly independent, you should really only see them co-occurring in about 4 in 10,000. But if they share something in their underlying biology, for example, atypical neural connectivity, maybe they would co-occur more often than chance. So we did this survey, 164 adults with autism, and we gave them a questionnaire asking about synesthesia. And what we found was that the rate of synesthesia in adults with autism, it's an online study, was about 3 times greater than in individuals without autism. So there seems to be a connection at the level of symptoms, if you like, or reported experience. Why might we expect autism and synesthesia to be linked? So there are a number of different ideas floating around, and they've all already surfaced in the conference. The first is the idea, bless you. The first is that autism might involve a reduction in long-range connectivity and an increase in short-range connectivity. You can see this image here that many of you would have come across from Matthew Belmonte and Nancy Minshew. But we've already heard the idea that this over-connectivity might also characterize synesthesia too. The second point here is that one gene that's underneath that linkage peak in our linkage study that we reported on chromosome 2 is a rare variant that's also associated with autism. So we might discover some genes that are shared by both conditions. And you can see in the bottom here this point about ERP, evoked response potential, that you can see at least in one study that if you test people with autism whilst they're listening to sound, they show interesting activity in visual cortex, suggesting that at an electrophysiological level we might be able to detect signs of synesthesia in people with autism. So it kind of raises this question about what might autism and synesthesia share. And so here I've just noted down some of the points made earlier this morning that seem to be common to both conditions. So we heard about upside-down face recognition superiority in synesthesia. Well, this has been known about for a long time in autism too. Some evidence of superior auditory discrimination, that study of DAR versus DAR, but which people with synesthesia might be better at discriminating. There's plenty of evidence that in autism that's also the case. Lots of discussion this morning about the balance between excitation and inhibition. Seems to be a popular theory of synesthesia. Well, if you look at the autism literature, it's very popular there too. And Caroline Robertson, who's going to be joining us later, may mention her interest in the GABA-glutamate ratio and measuring that in autism. Reduced pruning. Again, this is something that's discussed a lot in relation to autism, and we heard quite a lot about it in synesthesia. These two conditions may be much closer than we previously realized. In terms of sensory issues and why DSM-5 is now recognizing this as a core part of autism, we've been studying this, and this paper by Teresa Tavasoli, who left Cambridge and is now in New York at the Seaver Center in Mount Sinai, showed that actually sensory hypersensitivity is very common in autism and at much higher levels than in people without autism. And she's done this using various different tests, mostly self-report questionnaire studies, when we've tried to use psychophysics, it's much harder to get evidence for these differences, but at least at the level of what they experience, people with autism report hypersensitivity. And you can see here that when she looked for correlations between autistic traits measured on the AQ, the Autism Spectrum Quotient, and sensory hypersensitivity, you see quite marked correlations both in the autism group and in the typical group. This is a study which I found very interesting, which is now quite an old study in autism, but showing that if you put people with autism into an fMRI environment and play them just pure tones, you get greater activity in auditory cortex in the autism group than in the control group. So it's an imaging validation of what people with autism are telling us, namely that they experience the world more intensely. And we've known for a long time that people with autism also have better attention to detail. This is the embedded figures test. You have to find the target shape as quickly as you can in the overall design. People with autism are faster. This is the average number of seconds it takes to find the target shape compared to controls. And when you ask them to do that test whilst they're lying in an fMRI scanner, you find that people with autism show less activity in posterior parietal cortex compared to controls, even whilst they're actually faster and more accurate at doing the task. So this kind of task should probably be tried in synesthesia to see if we get similar results. So let's get down to the kind of neuronal level, because Eric Courchain in San Diego has been arguing for a long time now that there might be overconnectivity at the neuronal level in autism. So this is taken from his paper, but it shows development. In the typical case, you can see increasing connectivity across typical development. But in his post-mortem study, published in 2007, Courchain reported 65% more neurons in frontal cortex in autism compared to controls. So there's growing evidence for overconnectivity in autism. And this may well relate to a finding which is often reported but not without some failures to replicate, namely the early overgrowth findings in autism. So this is a Schumann study from the Journal of Neuroscience in 2010, showing that if you do repeat structural MRIs, the group in red are the kids with autism. They have larger total brain volume than the typical kids shown in blue at each of these ages. This is from 12 months old through to 60 months old, where each person gets a minimum of 2 brain scans, so you can join the dots to look at developmental trajectories. So more neurons, possibly, in autism, may be contributing to larger brains, and it's also reflected in larger head circumference in many studies. Here's another post-mortem study that people may be familiar with, again from autism. So this was published by Tang and colleagues in Neuron last year. We have a typical neuron on the left, and if we can call it an autistic neuron on the right. And what you can see, hopefully even with the naked eye, is that the autistic neuron has more white dots all the way along its length, and each of those white dots is a dendritic spine, or a location of a synapse branching to neighboring neurons. So this is again a suggestion from post-mortem studies that at least in autism we might be seeing evidence of overconnectivity. In the same paper, they were able to divide their post-mortem sample into the young versus the old, and what you can see is what they interpret as reduced neural pruning in autism, the dotted line, compared to the control group without autism. So maybe some evidence of reduced pruning causing the persistence of overconnectivity. Here's Courchain's work, Casanova's work, showing typical mini-column changes in the width of mini-columns as typical children get older. You can see they go from narrow through to getting wider in typical development, and this may well relate to Takeo's points about the critical period, and that in autism what you can see is that the mini-columns stay narrow in contrast to the wider ones that are seen in the typical group at later points in life. So maybe some evidence not just of reduced pruning, but also changes in the critical period in autism. I've mentioned the genetics of synesthesia briefly. Autism too is genetic. It runs in families, that where you've got one child with autism in the family, the likelihood of a second child also having autism is 1 in 3, compared to the general population rate of autism of 1%. So this is definitely telling us that autism is familial, and many genes have been associated with autism. So these are the genes that are kind of the strongest candidate genes from the Safari website, which you can see the URL at the bottom of that slide. They report every genetic association with autism. There have been probably over 500, but these are the genes which look like the strongest candidates because they've been replicated. But we know that autism isn't just genetic. So these girls are identical twins, but one has autism and one doesn't. So the very existence of twin pairs of this kind, given that they're genetically identical, must mean that autism can't be wholly genetic. And these days there's a lot of interest in epigenetic factors that might be acting on the gene to change gene expression, protein levels, and indeed some of the genes on this list show differences in methylation between the affected and unaffected twins where we have these discordant pairs. So I wanted to move to the whole issue about gender and sex-linked biology, because certainly autism, as many of you know, is associated with being much more common in boys than girls. This is data from the Center for Disease Control, so it's kind of the most recent epidemiological data on autism. And what you can see is that autism is about 1 in 68 in 6 different states where it was studied. This is a school study, school-age study, but you can see it was 1 in 48 boys compared to 1 in 189 girls. So this slide also shows you that autism is associated with low IQ as well as average or even above average IQ. And this slide kind of captures the whole autism spectrum, but what's very clear is the link with gender. The link with gender could, of course, just reflect that maybe we're not very good at detecting autism in females. Maybe our diagnostic methods are not sensitive to it. But it could also reflect something about underlying biology, causal factors. So we've been looking at the biology that might influence autism risk in males more than females. So here I take a slight detour into just reminding you about genetics and hormones and how they can change not just the reproductive system, but also the brain and the mind and behavior. So you'll all have heard of the 4 core genotypes model, but essentially you've got the sex chromosomes here, and what you can see is the XY genotype or karyotype for a typical male, which has the SRY gene. It's sometimes referred to as the TDF or testes determining factor on the Y chromosome, and when that is present, it gives rise to the growth of the testes, which produces testosterone, and you get the male phenotype in terms of the reproductive system. And of course you've got the female genotype, 2 X chromosomes, and in the absence of that SRY or TDF gene, you get the production of the ovary, the growth of the ovary, and you get the female phenotype. Well, what's been possible in animal research, particularly in mice, is you can move that gene onto an X chromosome, and what you find is that an animal that's chromosomally female can grow testes, produce testosterone, and end up with a male phenotype in terms of the body. Or you can do the opposite. You can delete the SRY gene or TDF gene from a chromosomally male mouse, and the result is that the animal will grow an ovary and end up with female phenotype. So this is the so-called 4 core genotypes that you can start to disentangle the role of genetics, or at least sex chromosomes, but in particular the SRY gene, from the role of prenatal androgens, testosterone being the clearest example. The early classic experiments involved really quite blunt instruments to manipulate hormones in rabbits in this case. So back in the 1940s and 50s, you could take a male rabbit, use castration, so depriving the male animal of his source of testosterone, and find that the animal developed a feminized reproductive system. And you could do the reverse experiment. These were done by Jost, and where you could take a female rabbit, for example, implant a testes so that she is exposed to male levels of testosterone, and her reproductive system is masculinized. So this was showing very early on that these prenatal hormones were very important in terms of the development of sex, particularly around the reproductive system. But these hormones also seem to change the brain. So another way to manipulate the hormones is not to do something surgical, but simply to give, for example, a typical female rat, a lot of this work is done in rodents, extra testosterone and see what happens to the brain. So if we look at a particular part of the brain, the SDNPOA, sexually dimorphic nucleus of the preoptic area in the hypothalamus, you can see this is a typical male, and here's a typical female. These are obviously average sex differences. But if you give a female rodent extra testosterone, her SDNPOA becomes as big as a typical male. So these were kind of early proof of concept demonstrations that prenatally the hormone testosterone is changing the brain. You can see this very clearly in these studies looking at volume of that region, the SDNPOA, where you've got age of the animal, and you administer either oil as, if you like, the placebo, or the inert substance, or testosterone, and you're looking almost day by day during gestation to see what's happening to the volume of this particular brain region. And under the influence of testosterone, you can see that suddenly volume of the SDNPOA shoots up around day 18 of gestation. So very clear evidence that the hormone is changing at least one part of the brain. That part of the brain turns out to control mating behavior. So Phoenix in classic studies showed that if you, again, manipulate the hormones, either by giving a typical female testosterone treatment or taking a typical male and using castration, you can change mating behavior mediated through that brain region from a typical male to a typical female pattern of behavior. And without manipulation, you can see that this is the normal male and the normal female pattern of behavior. So the hormone prenatally is changing brain structure, but it's also changing behavior. So this is my most exciting slide, because here we are back in that same brain region, SDNPOA. And here we're looking at the rat brain, and we're looking at typical female in terms of neurons in the SDNPOA. And then when she's given extra estradiol, an estrogen is aromatized from testosterone, so it's in the same pathway, and also has this masculinizing effect. And what you can see is that you get much more connectivity in that brain region under the influence of extra sex steroid hormone. There have now been studies in humans, jumping from animals to humans, looking at sex differences in cortical synaptic density and showing differences between males and females. So this hormone may well be relevant in the human case to synaptic density. But just going back to the animal work, where you can look at this in with much more experimental opportunity. If you administer the hormone, shown in green, compared to an inert substance, oil, shown in beige here, and look at the total number of cells undergoing apoptosis or pruning in the SDN-POA, you can see there's reduced pruning under the influence of testosterone in these male rats that were castrated at birth, but where you reintroduce testosterone artificially. So the hormone seems to be changing apoptosis, and maybe this is very important for how we're thinking about synesthesia and autism as an alteration in rate of apoptosis or pruning. There's one more way that you can manipulate the hormones, and that's a much more natural way, and that's where you have animals that have litters. So this is the so-called twin transfer method of looking at the effect of testosterone. So in the case of rats, for example, often a mother rat has many offspring in one litter, and you can look at female fetuses that are either adjacent to one female, two females, or a male brother, because in those instances the female will be exposed to more testosterone. So here we've got these little rat fetuses. This one here is called a zero male. She's female, but she's got no male on this side. This one here, no male on this side. This female is called one male because she's got a female next to her, but a male next to her. And this female is called a two male female because adjacent to her she's got a brother on each side. So it's a natural way to look at the dosage effects of testosterone on brain behavior. And well, here we're just looking at the reproductive system, because you can see that depending on your location in the litter, this measure of the reproductive system, the anogenital distance, you can see has a typical sex difference, the average female and the average male shown in blue. But depending on your position in the litter, you can see that a female who's surrounded by two male brothers has a much longer, more masculinized reproductive system compared to her sisters. So the hormone is affecting somatic changes in the body. So let's move now to humans, and to tell you a little bit about our work in looking at this hormone in autism. And it's actually, again, a sort of double pleasure for me to be in Boston, because the name Norman Geschwind will be familiar to many of you, who developed the hypothesis that maybe prenatal testosterone was important in a range of human developmental conditions, autism, language delay, and other conditions that are more common in boys than girls. Geschwind was a neurologist, and he died before the advent of amniocentesis, which was introduced in the 1980s and became clinical practice in the 90s, which is when we got interested to study this. So amniocentesis is kind of an opportunity where you can measure in humans in an ethical way how much testosterone is in the amniotic fluid surrounding the human fetus. Geschwind's big idea was that testosterone has organizational effects. Organizational is another word for permanent, that the amount of testosterone you're exposed to as a fetus, it's a critical period of a different kind, will permanently change your brain structure and later brain function and behavior. So you can see here that you can measure testosterone in the amniotic fluid of male and female fetuses in women who opt during pregnancy to have amniocentesis, where a needle is introduced into that fluid, and then you get to measure the individual variation in testosterone. Boys shown in blue, girls shown in red, but look at the huge individual variations. Some boys are so low in testosterone they're in the female range. Some girls are so high in testosterone they're in the male range. But we were very interested in a study of about 600 babies whose mothers had amniocentesis to look at individual differences. What does it predict about your later development? So we followed up these babies. We were looking at that surge in testosterone in males, which happens prenatally, here's birth. There's a second surge in testosterone that happens soon after birth. And of course, there's a big one at puberty. But we were just measuring how much testosterone you have prenatally in just over 600 kids. And we tested them at various different points. This is when they were 8 years old and they could come into our MRI scanner just to demonstrate that what you find in animals you also find in humans, namely correlations between how much testosterone the child was exposed to prenatally and changes in grey matter volume in different parts of the brain. So correlations both positive and negative with prenatal testosterone. But when they were 8 years old, we were also able to give them this test of attention to detail. How quickly can you find the part within the whole? This is a child version of the same thing. People with autism, as I said earlier, are super quick and super accurate on these tests. But what we found was that prenatal testosterone positively correlates with your performance on this attention to detail task in typically developing kids. We also were able to ask the mothers to administer a measure of autistic traits in these kids. 600 kids is way too small a sample to look at correlations with diagnosed autism if it's only 1% of the population. So instead we looked at a dimensional measure of autistic traits. And again, what you see is that the higher your level of prenatal testosterone, the more autistic traits the child is showing. This was at 4 years of age. And then just earlier this year, we were able to finally demonstrate a link between prenatal testosterone and other sex steroid hormones in this delta-4 pathway. You can see testosterone is at the bottom of this pathway, but these are other androgens that each one is synthesized from its precursor. So we measured all of these, but here I'm just showing you the testosterone result, but it was true for all of these sex steroids were elevated in kids who went on to develop autism compared to typically developing kids, no autism. And that was only made possible through a collaboration with the Danish Biobank. In Denmark and Copenhagen, they've been storing amniotic fluid from amniocentesis since the 1980s. They have over 100,000 samples in their deep freeze. And they also have a register for who's developed autism. So we were able to know who's got autism, look back in the freezer to see if their amniotic fluid was available. And on that basis, show that prenatal testosterone may be just one part of the story of the development of autism. For me, prenatal testosterone is very interesting for other reasons because it has epigenetic properties. We saw earlier that autism is genetic, but there's likely epigenetic factors involved. And testosterone is one potential epigenetic factor. But the amount of testosterone a person produces is also mediated by genes that control that, or that operate in the synthesis of testosterone, like CYP17 and HSD17B2. And both of those genes have been associated with autism. I'm going to finish up with just mention of an animal model that was published just this year from a Chinese group where they manipulated testosterone in rats and gave them a social test, whether they wanted to spend more time with another rat or in an empty cage. And using this drug, letrozole, which blocks aromatase, so it causes a buildup of testosterone. Less testosterone is being converted into estradiol. What they found was that the animals who had higher testosterone because they were on this drug showed, if you like, reduced social interest in a conspecific. So this is maybe the first animal model evidence that is linking with our human work. So Takeo talked quite a lot about GABA and GAD67 as the driver of inhibition and the critical period. And so whilst he was talking, I was not being rude, I was Googling whether the sex steroids interact with the GABAergic system. And this is a study which I found in goldfish, but there's a whole literature on how GABA and the prenatal sex steroids interact. So I think we should keep in mind that whatever is influencing changes in the critical period, and GABA is a key player, might also interact with the sex steroid hormones. Here's my conclusion slide, I hope I'm on time, that we've seen evidence that autism and synesthesia occur more often than chance. Maybe both of them entail atypical neural connectivity, overconnectivity. Maybe they both share reduced pruning in early development. They may also both share certain perceptual or cognitive features, and this really needs unpacking, but there was the perfect pitch potentially as an overlap and better attention to detail, and some unusual performance on things like upside down faces and so forth. And maybe both of them involve alterations in the excitation inhibition GABA glutamate ratio. I hope I've shown you that the prenatal sex steroids, such as testosterone, seem to be important in these kind of phenotypes, and it now linked to autism. And I think going forward, it will be very interesting to test the role of these prenatal sex steroids in synesthesia. And I'd just like to thank my funders. Thank you. Do you have time for a couple of questions? Yeah. Sure. Yeah, go ahead. Do you want to wait for the...
Why are girls and boys equally likely to have synesthesia when the boys have different levels of autism? And then I have another question, which is sort of a follow-up question. So much synesthesia depends on personal rapport, and I wonder if somebody who's hypersensitive to pattern has simply got a lower threshold for discerning their own synesthetic responses, as opposed to having literally more synesthetic activity.
Right. So I think we're kind of limited in how we can identify either of these conditions, but especially synesthesia is kind of, it hinges on self-report. So we don't really know whether the epidemiology, the prevalence rates, and particularly in males and females, how accurate they are. When I started off in synesthesia research, all of the studies were showing an excess of females compared to males. In our own early studies, 2006, we had a population study, 8 females for every 1 male. So we thought that synesthesia was sex-linked, but in the opposite way to autism, much more common in females. I heard earlier at the beginning of the morning this claim that synesthesia is now no difference between males and females. That may just be how we've found ways of recruiting, but in a way, I think what the work in autism has generated is it doesn't really matter what the sex ratio is. It was just a way to start thinking about sex-linked biology and to go deeper into it. So the old days, autism was seen to be about 4 males for every 1 female. These days, it's more like 2 males for every 1 female as we get better at identifying autism in girls. But either way, it was just a handle to kickstart research into looking at sex-linked biology. Other questions? There's one at the back, yeah.
You pointed out all these similarities between autism and synesthesia. I was wondering, these are pretty much sort of high level, even the biology-related ones. I was wondering if there are sort of indications or some findings that they share some molecular mechanisms as well. If one, for instance, is a mild form of the other, or
yeah, I mean, I'm hoping that one output of today is that we will start trying to mirror some of the work that's going on in one field in the other. So we can start looking at maybe common shared biology between the two. So I would say that the comparison between the two conditions is still very much in its infancy. Yeah, do you want to wait for the microphone?
You know, one comment is that did people measure those, sex hormone level in high-performing autism group across different age groups,
right,
versus those, they are low performance, or they have different ASD, because not all are created equal. Some of the Rett syndrome, they have different level endorphin, and in the tons of literature, they also look into the endorphin level.
Yeah.
They might be lower in certain group, might be no change, and
sure.
I think it's a garden variety of diversity. That's what I think.
Sure. There is a lot of diversity in autism, so I'm glad you mentioned it. I showed you that one slide from the CDC, which kind of, in a snapshot, showed you the diversity. That autism is associated with any point on the IQ scale. And we could do the similar sort of picture with language too. Some people with autism have no language. Other people with autism are hyperverbal, precocious language. When we found that link between prenatal sex steroid hormones and autism, that was across the whole IQ and language range, which surprised us. So it wasn't just the Asperger group, it was also seen in individuals with classic autism, which could include learning difficulties too. So I'm sure you're right that we need to subgroup, that some of these findings may not apply to every subgroup, and the so-called syndromic forms of autism, like Fragile X, may have a completely different etiology, although even there, we might expect common pathways.
Yeah, my last quick comment is that if you look into a deeper molecular level, the progesterone or the derivative, they might affect, the intracytoplasmic domain of the GABA receptors, and that, very well known, among in the Boston Medical Center Research Group.
Okay, thank you for that. I mean, I think I was kind of making that point that the sex steroid hormones interact with the GABAergic system and maybe GABA receptors, and that we all tend to study different parts of biological pathways, GABA, sex steroids, but actually they do interact.
More questions? So the similarities you pointed out lead one to wonder whether some of the interventions Cal talked about for taking off the brakes in order to rebalance excitatory and inhibitory mechanisms might be effective therapies in autism. So I'm wondering if there has been any approach using any of those ways of taking the brakes off and rebalancing excitation and inhibition that he talked about?
I mean, Takeo might know the answer to this, but my impression is that the basic research is ahead of the kind of treatment research in this area. Is that right?
I think the ideas are there, and we need opportunities to do that. So Michela Fagiolini, who was here, has worked a lot on the MECP2 mouse model of Rett syndrome, made some very interesting observations about hyper-mature inhibitory circuits, the critical periods are earlier, and then intervened to slow down the maturation. And this seems to prevent a large part of the regression, at least in the visual domain. And we're working on other ways to do this more globally to see if it rescues other aspects. but it's still in a preclinical stage. Sometimes you get lucky. Drugs that are used clinically for other purposes, like ketamine, seem to have an effect in this direction in the mouse model, just published this year, a few months ago. And so hopefully that kind of opportunity would fast track this kind of approach. Yeah. I have one question since I'm holding the mic. I suppose the Danish database is so thorough that they've also documented synesthesia in there.
So the Danes use, they don't use DSM, they use ICD. So it's the World Health Organization classification system. Synesthesia actually doesn't pop up in either DSM or ICD. Right. It's not technically a psychiatric condition, it's just a difference. And as you know, many people with synesthesia don't complain of having synesthesia. They don't feel it causes suffering. So it doesn't, in that sense, warrant a diagnosis. And so that's why it's not in the Danish register.
Of course, I was only joking, but if you revisit those autistic subjects and if you have a chance to measure synesthesia, that might be very interesting.
No, that would be very good. Unfortunately, in the Danish register, there's ethically there's no recontacting. So their biosamples are there from prenatal, and it includes things like maternal serum, but no opportunity to recontact the kids who some of whom may now be adults. So it's all anonymized.
It raised a tangential question in my mind. Is there some epidemiology globally that certain cultures have more synesthesia, maybe if they use more visual-based language, I don't know, some kind of influence that might be related to that?
I don't know the answer to that. Does anyone else who's here amongst the synesthesia researchers? I'm not sure that there's been much in the way of cross-cultural studies. Daphne wants to comment.
This is elaborating on the answer I don't know. There have only been 3 studies to get population estimates of the prevalence of synesthesia. They were done in science museums and first-year psychology classes. So they're population-based studies, and that's where the 4 to 5% incidence and as common in males or females come from. But that's been done only, actually, I think I can say only in Britain. Anyway, only in the West, so we don't know.
Yeah, and it doesn't really add up to, to epidemiology. They're not representative samples.
They're not representative samples, but they're more representative than what we had before, which were people who volunteer to go in the lab when you advertise for synesthesia.
Okay.
One last question. Given your talk about sex steroid exposure in utero, is there a correlation with environmental changes affecting this and the incidence of autism rising that you know of?
Well, as you know, there's a whole field of endocrine disruptors, which is basically environmental factors that can change our hormone production and hormone exposure. I'm not aware of any work looking at how that might link to autism. And I think we have to be quite cautious, just because we don't want to raise anxiety that there might be environmental factors that could be causing autism in the absence of strong evidence. But you're right that autism has become more and more common. So when I started off in this field, autism was thought of as quite rare. 4 in 10,000 was the textbook figure in the 1970s. Now it's 1%. But most of that kind of increase is probably attributable to better recognition and broadening the diagnosis to include Asperger's and having more services on the ground. So more eyes looking for it.